Good evening, dear students. I hope I'm audible. You can see my screen also. Please let me know. If all fine, please let me know. Today we are discussing with the topics that is MCQs of the oral histology and dental anatomy. We'll be discussing that. So here we will be mainly focusing on the important MCQs, the concepts which you will understand and which will be helpful for you to solve the different MCQs for the subject of dental anatomy and oral histology. So dental anatomy is the one where you need not understand any concepts here. It's just a simple memory based dental anatomy. Whereas oral histology requires certain imagination, certain things, concepts which have to be applied. Okay. I hope my voice is clear. You're able to hear me. Let me start with the first MCQ here. The first MCQ is that the differentiation of the ameloblasts from inner enamel epithelium takes place during which stage? That means in directly, rather than asking directly, histodifferentiation. What is histodifferentiation? Histodifferentiation means conversion of the cell into another cell. The inner enamel epithelial cell will become ameloblast. That is called as histodifferentiation. The dental papilla cell will become odontoblast. That is called as histodifferentiation. So when the histodifferentiation takes place, the histodifferentiation phase takes place or corresponds to the early Bell stage. Okay, so the question is that differentiation of the ameloblasts from the inner enamel epithelium takes place during which stage? During the early Bell stage. That means histodifferentiation corresponds to the early Bell stage. Same way, if a question is asked like the differentiation of odontoblast from a dental papilla cell takes place during which stage? Takes place during early Bell stage. Okay, so this is what you have to remember, that histodifferentiation occurs during early Bell stage. Okay, am I clear? My voice is clear? You able to see the screen? Please type it in the chat box. Okay, so here basically in the physiological stages of tooth development, we know the physiological stages of tooth development: initiation, proliferation, histodifferentiation, morphodifferentiation, and apposition, right? So we have 5 stages, physiological stages. These physiological stages basically we have to understand they correspond to the particular morphological stage. Okay, so we should be able to understand which stage, physiological stage, corresponds to which morphological stage. Okay, so that is important, you have to remember. So histodifferentiation occurs during early Bell stage. Okay, moving on to the second question. During the early illness of the child, the tooth calcification is disturbed. That means calcification is disturbed. Calcification means hard tissue formation is disturbed. This will most likely affect the tooth in which stage of development? So in other way, they are asking hard tissue that is formed, right? So occurs hard tissue formation, that is enamel and dentine, occurs in which stage? Occurs in late Bell stage. So till the bud, cap and early Bell, there is no hard tissue formation, no enamel, no dentine. Directly in the late Bell stage, you will see enamel and dentine form. First dentine forms and then the enamel forms. Okay, this is very important. So that is why you have to remember that till the early bulb stage, the ameloblasts will receive the, till the early bulb stage, the ameloblasts receive the blood supply. They will receive the blood supply through dental papilla. Whereas in the late bell stage, the ameloblasts will receive the blood supply from dental sac, because a hard tissue is already formed. In late bell, you see enamel and dentine formation, whereas hard tissue formation has occurred. So obviously, blood cannot flow, nutrition cannot reach the ameloblasts. Hence, the ameloblasts will depend upon the dental sac capillaries. So dental sac capillaries. Okay, so this is what you have to remember, that late bell stage, the tooth calcification is affected. Okay. Ossification of mandible starts in which of the following regions? We should understand that basically mandible is one bone which forms by intramembranous way. Intramembranous. Majority of the bone mandible occurs by intramembranous way. Okay, so you have to understand that there is something called as Meckel's cartilage. There is something called as Meckel's cartilage, and this Meckel's cartilage is related to the nerve. We have a nerve here, right? So that nerve that forms basically is the mental nerve and incisive nerve. So mental nerve and incisive nerve will divide here. This is basically the inferior alveolar nerve. So, and what is this? This is Meckel's cartilage. Now what happens is basically at the junction of the incisive and mental nerve, within the membrane, there will be a membrane here, there will be a membrane here, and in this membrane basically, which is there around the Meckel's cartilage, here you will see the ossification. You will see the ossification here. So first ossification that takes place is in the region of mental foramen. So it starts here. So what you have to remember, Meckel's cartilage will be there. There will be a membrane around that. So where the inferior nerve will divide into incisive and mental nerve, there the first site of ossification will appear. Remember, the first site of ossification occurs 6th week of intrauterine life. 6th week of intrauterine life, you will see the first site of ossification. That is why majority of the mandible, let it be body of the mandible, let it be ramus, is formed by intramembranous way. Only the symphysis part, condyle and coronoid are formed by endochondral. So three parts of mandible which are formed by endochondral ossification is symphysis, condyle and coronoid. Rest body of the mandible and ramus of the mandible is formed by intramembranous way of ossification. So this is a very important question. You have to remember, sometimes they'll ask you, is Meckel's cartilage directly related to mandible formation? No, Meckel's cartilage is not directly related. It just acts as template. Acts as template. Template means it acts as a framework. Okay, that is what you have to remember. Okay, next. During the cavity preparation, a dentist drills deeply into the tooth, exposing the pulp cavity. He covers the exposed pulp with calcium hydroxide. So there is a pulp exposure. Now pulp exposure has been covered with the calcium hydroxide, and the dentist reassures the patient. Which of the following cells of the pulp are most likely to be responsible for healing and repair? Okay, for this you have to understand that the undifferentiated cells which are there in the pulp, these are the major cells. Okay, these are the cells which are going to help for the in healing and repair. So these undifferentiated cells will be there in your pulp core. They will be there in the pulp core and cell rich zone of pulp. So pulp has got two zones where the cells are concentrated. One is cell rich zone, one more is a pulp core. So in those both of these areas you will see these undifferentiated mesenchymal cells. These undifferentiated mesenchymal cells are responsible for repair. Whatever repair occurs is because of these cells. Okay, so we have to remember that where are these cells concentrated and what is their main function. Okay, which of the following is the last succeedinous tooth to erupt? Last succeedinous tooth means the last tooth to erupt in the oral cavity. Permanent tooth, succeedinous tooth. Okay, like we don't have certain teeth which are without a succeedinous. Like for example, they don't have any predecessor. They don't have any predecessor. Like basically your first molar, second molar, third molars, they don't have any predecessor. Okay, so last succeedinous tooth to erupt into the oral cavity is maxillary canine. Maxillary canine. So here we have to remember the order. All of us know that the main difference in the eruption between maxilla and mandible is between canines and premolars. In maxilla, the premolars, maxillary premolars erupt first and then the maxillary canine will erupt. Whereas in mandible, mandibular canine will erupt first and then the mandibular premolars will erupt. So that difference we are supposed to know between the eruption in the maxillary and the mandible. Okay, so sequence of eruption is a very important topic, very important question they can ask you. And chronology. Chronology is also again a topic for very commonly asked single liners. Like when is the first, when does the initiation takes place of that particular tooth? When does the first evidence of calcification takes place? When does the root formation complete? When does the eruption take place? All those four things are asked usually. What are the four things? When does a tooth initiate? What is the first evidence of calcification? When does the tooth erupt? When the root completes. So four things are usually asked. So you are supposed to know all these four things for every tooth. For every tooth. Most poorly developed cusp in maxillary second molar. So if you see here, maxillary molars, the largest cusp is mesiolingual cusp. All of us know that this is the largest cusp, mesiolingual cusp in maxillary molars. Okay, and this mesiolingual cusp only, on this cusp only you will see cusp of Carabelli. Cusp of Carabelli, that is the fifth cusp, is present on this mesiolingual cusp only. Okay, so this is the largest cusp, and if you see the smallest cusp being the distolingual cusp, that is the smallest. That is the smallest. Okay, so if you see from transition from first molar to second molar, the distolingual cusp becomes small or it is absent in second molars. So in maxillary second molar, this distolingual cusp is either small or it is totally absent. And if you go to third molar, it will be definitely absent. So third molar will have only three cusps: mesiolingual, mesiobuccal, and distobuccal. Obviously, cusp of Carabelli will also be absent. So cusp of Carabelli will be present only in the maxillary first permanent molar. Permanent molar, that too maxillary first molar. In the deciduous, cusp of Carabelli is present on second molars, maxillary second molars. Okay, so you have to remember that cusp of Carabelli is a feature of permanent maxillary permanent first molar and deciduous maxillary second molars. And distolingual cusp is the smallest one. It disappears or it becomes a small in case of second molar, maxillary second molar, and is definitely absent in case of maxillary third molar. A Y-shaped occlusal groove is most likely to be present in. So which premolar will have a Y-shaped groove is mandibular second premolar will have Y-shaped groove. So we should remember that mandibular second premolar is one particular tooth which can be of two types. It can be of two types. It can be a 2 cusp type or it can be a 3 cusp type. Okay, 2 cusp type or 3 cusp type. What do you mean by 2 cusp type? It will have one buccal cusp and one lingual cusp. Whereas here it will have one buccal cusp and two lingual cusp. Two lingual cusp. So in the 3 cusp type, what happens is usually, so suppose this is the buccal, this is a groove. Okay, this is the groove that you see. In the this one. Now what happens is basically it will have two lingual cusps. So that will have typically a Y-shaped groove pattern. Y-shaped groove pattern. In case of 2 cusp type, either it will be U or it will be V. So we are supposed to remember this groove pattern. This groove pattern is very important. Where do you see U or V? Where do you see Y groove pattern? It all depends upon whether it is a 3 cusp type or whether it is a 2 cusp type. So this is a peculiarity of the mandibular second premolar. Remember, it can be a 2 cusp type or 3 cusp type. Depending upon the cusp type, the groove pattern will be there. Either it will be Y or it will be U or V. Okay, so this is very important regarding the mandibular second premolar. This is a very frequent MCQ, very commonly asked MCQ regarding the groove pattern that you see. The roots of which permanent tooth are normally formed by the age of 10 years. See, there is a rule here. I have told you how to answer such questions. We can't remember right everything. We know about the eruption. Generally remember, permanent tooth eruption is, suppose, is some age. We know some age eruption, that X, like that is considered X as the age. Root completion for permanent at least minimum X plus 2 years. Minimum, I'm telling you, 2 to 3 years. 2 to 3 years. Root formation occurs X plus 2 to 3 years. Like for example, here they have given that root completion is occurring by 10 years. Okay, so usually by if we minus here 10 years and here we have X plus 2 years. If we consider, so X will become 10 minus 2 years, that is somewhere around 8 years. And which is the tooth which erupts around 8 years? It is upper central, 7 to 8 years. 7 to 8 years. So this way you can solve the questions based upon the root. Generally we have to remember either it is 2 to 3 years. Some books even give just mean as 2.5 years. For permanent tooth it is 2.5 years after root completion. So it will be somewhere 7 to 8 years. So if you take the range will be 7 to 8 years. So which tooth erupts during 7 to 8 years is upper central incisor. Upper central incisor. Okay. Now going on to the next question. Histology of enamel can be best observed using which of the following methods of slide preparation? That means we know that we have to study the enamel. Enamel is a hard tissue. We know that hardest tissue. Okay, so we have to study this enamel. Okay, the best way to study enamel is always through ground section. To make a ground section is the best way. Okay, ground section usually will have average of 50 microns thickness, 50 micron thickness, and we can, based on that, based on different tissue which we want to appreciate, there will be different thickness. But remember that ground section is the best way to study enamel. to study enamel. Let it be enamel structures like incremental lines of enamel. Let it be to study the enamel tufts, enamel lamellae, enamel spindle. All those things you will better understand in the ground section. Obliteration of dentinal tubules by minerals will result in. So we know that when the dentinal tubule, normally S-shaped dentinal tubule which is seen, this S-shaped dentinal tubule is filled with basically is containing odontoblastic process. So instead of the odontoblastic process, if lot of calcifications takes place here, there is minerals are deposited here, right? So if this minerals deposited, what happens is the dentine, the refractive index of this dentine becomes the same as that outside. So Ri here and Ri here becomes the same. So what happens is the dentine, such a dentine is called as transparent or sclerotic dentin. So transparent dentine or sclerotic dentine is a particular age change. Please remember, it occurs with age. So where what happens is dentinal tubule will be filled with minerals, hence the refractive index will become same, hence the dentine becomes transparent or translucent. Remember, as it is the minerals are deposited, this dentine has got a higher noob hardness. It is around K80. Normally dentine has got 68 noob hardness number. Okay, normally dentine has got 65 to 68 noob hardness number, whereas sclerotic dentine is much more harder than the normal dentine. So sclerotic dentine, the hardness will be somewhere around 80 noob hardness number. Okay, again this is a very important MCQ that you should know about the hardness numbers. Let us move on to the next question. A 12-year-old boy presents to the dentist with his broken maxillary central incisor, of which the enamel is chipped off with the exposed dentine, and he complains of pain while drinking the cold water. Which of the following contents of dentinal tubule are most likely to be responsible for the pain felt by the patient? So indirectly they are asking that, rather than directly asking, the main reason for dentinal pain hypersensitivity. So dentinal hypersensitivity is expressed by main three theories. We know one is called as a direct nerve stimulation theory, one is called as the hydrodynamic theory, okay, one more is a transduction theory. So among the three, the hydrodynamic theory is the most accepted. According to this theory, the dentinal fluid which is there around the odontoblastic process, this fluid will move. Because of this fluid movement, the nerve endings will be stimulated, that leads to pain. So the main here is the fluid. So that explains the hydrodynamic theory. That explains the hydrodynamic theory for dentinal hypersensitivity. Dentinal hypersensitivity. So this is the most accepted theory. Most accepted theory for dentine hypersensitivity. Which of the following has the highest mineral content? If you see the peritubular dentine, like for example, this is the dentinal tubule. Suppose these are the dentinal tubules. The peritubular dentine, when I say peritubular dentine, the dentine which is there around the dentinal tubules, this yellow color one. This yellow color one, this is more mineralized. Its mineralization is almost 9% more. This peritubular dentine, this is almost 9% more mineralized. This is 9% more mineralized than the intertubular dentine. So this is very important, remember. So rest all whatever we see, this is your intertubular dentine. This is your intertubular dentine. So peritubular dentine is more mineralized. The mineralization status is almost 9% more than that of the intertubular dentine. Okay, this is what you have to remember. So peritubular dentine is highest mineral content. Highest mineral content. Okay, let us move to the next question. A 20-year-old man falls down and chips the incisal edge of his maxillary central incisor, reducing the length of the crown. That means crown has reduced length. A dentist informs him that the tooth may erupt a little to compensate for the loss. The tooth movement is likely to result in increase in the size of which of the following dental tissues? So we know that cementum is the one which will be deposited. For example, if this is a tooth, if this is the tooth, right, and if suppose this part, if this part gets chipped off, so what happens is there will be deposition of the cementum here. So because of this deposition of the cementum, the tooth elongates, okay, and erupts so as to maintain the occlusal plane. So that might slightly result in the eruption of the tooth, and that occurs mainly because of a cementum. So if this deposition occurs more sometimes, you will call it as hypercementosis. Which is a pathology. You should remember that hypercementosis is excessive deposition of the cementum, which can be localized or generalized. This is localized, what we have discussed. That is to compensate the occlusal plane. Okay, occlusal plane. Generalized can take place in case of Paget disease. There is something called as Paget disease of bone. In this Paget disease of bone, you will see generalized hypercementosis. Okay, moving on to the next question. A Deepcarious lesion reaches the pulp. The pulp responds by vasodilation. There is migration of WBCs into the pulp. This response is most likely to be initiated by the cell in which of the following zones? So that means indirectly they are asking which is the zone where blood vessels are there. So the pulp core is a zone where you will have nerves and blood vessels. So there will be nerves, major nerves and blood vessels in this layer. So those blood vessels will contain basically the WBCs, and those WBCs will migrate to the site, okay, to the site where there is the inflammation. Because of caries, they will spread, they will go and migrate there, and they will create, and there will be inflammation, pulpal inflammation. So main, you should remember that the main zone from where this is all this response is initiated is pulp core, because pulp core is the one which contains the blood vessels and nerves. Okay, let us move on to the next question. Dentine which is produced continuously throughout life is, we know that dentine is of three types primarily. We have primary dentine, right, and we have secondary dentine, and we have tertiary dentine. The one which is formed before root formation is primary dentine. One that is formed after root formation continuously in the life, throughout the life, continuously that is deposited is tertiary dentine, is secondary dentine. Tertiary dentine always occurs after stimuli. Whenever there is any stimuli, then only you will see tertiary dentine formation. Otherwise, there will not be any tertiary dentine formation. So, secondary dentine that is formed continuously. And remember, secondary dentine is usually intratubular. So what happens is, suppose this is the tubule, this is the dentinal tubule, it will get deposited continuously here within the tubule. So intratubular dentine formation is the one which occurs continuously throughout the life. So if they ask you option like primary, secondary, then tertiary, then the answer is secondary, which occurs throughout the life. And if they ask you intratubular, then the answer is intratubular. So you should be prepared for both. Okay, they can ask you anyway, so you should know the answer. Either it could be secondary or it could be intratubular dentin. Okay, moving on to the next question. During the process of tooth development, child suffers from febrile illness, because of which the tooth development temporarily slows down and then resumes normal soon after. In which of the following histological changes feature this change is apparent? So there is some febrile illness, there is some illness, fever. So what happens is, there is one structure called as incremental lines. So those are the strands which are going to represent the rhythmic deposition, right? Either it could be enamel, let it be dentine, let it be cementum, let it be bone. So in case of the dentine, we have lines of von Ebner. So what happens is that lines of von Ebner will become irregular and the distance will vary between the two. Normally, enamel and dentine are deposited in the rate of 4 microns, 3.5 to 4 microns per day. So in between each day, you will see one line that is nothing but incremental line. So because of the fever, because of the illness, the lines of von Ebner will be abnormal. Lines of von Ebner are the incremental lines of the dentine. So these are the incremental lines of dentine. Incremental lines of dentine. So we should be knowing the basic concept. How does the incremental line form? How does the neonatal line form? Okay, neonatal line forms at birth, exactly at the birth. That is why all the deciduous teeth and first permanent molars will have neonatal line, because first permanent molars are initiated at birth. So obviously the first permanent molars also will have the neonatal line. First evidence of calcification of maxillary permanent central incisors occurs at, we know that usually it occurs as 3rd to 4th month. So this is again a simple memory based question. That's what I've told you. First evidence of calcification is also a very favorite topic, common topic. Initiation, first evidence of calcification. So remember, initiation of all the first permanent molars occurs at birth. This is very favorite. Remember, all permanent first molars, if you take all permanent first molars, if you take, they are initiated at birth. You should not forget this sentence. That remember, they are initiated at birth. Okay, rest all initiation will take place later, but only at the first permanent molars initiation occurs at the birth. So this is again a very favorite topic. Okay, let us move to the next question. In salivary glands, the ducts that are lined by single layer of tall columnar cells with radially arranged mitochondria. So in other way, they are asking which is a duct which has got mitochondria. We know that there is a duct called as a striated duct, where its surface will be striated like this, and in between the striations you will see mitochondria here. You will see mitochondria here. So this is a striated duct, this is a striated duct, and this striated duct will have these mitochondria. So this folds, these basic folds and these mitochondria are responsible for this striated appearance. So striated ducts is the answer. Why mitochondria is there? The simple reason, mitochondria produces ATP, and we know that here whatever ion exchange that takes place in the saliva is against the concentration gradient, hence it requires mitochondria. Okay, this is again a simple memory based question. During the early stages of tooth development, 3 transitory structures can be seen. Early stages means only during cap stage, remember. So only during cap stage of tooth development you will see some transitory structures. Those transitory structures are enamel knot, cord and navel. One more that is remaining here, that is not given in the option here, is enamel septum. So we should remember the order here. Always I have told you, we have to remember the order. Always enamel knot will become a cord, and a cord will become a septum. Cord will become a septum. Septum will meet which part? Septum will meet the navel. Septum will meet the navel. So we should remember, we should be aware of these transitory structures during cap stage. The main function of these transitory structures is they are reservoir cells. They are reservoir cells. So whenever any damage occurs or any requirement of a particular type of cell is there, these cells can differentiate. That is why we have to remember these transitory structures. Okay, let us move to the next question. Concerning the stratum intermedium, one of the following statement is incorrect. It is composed of 2 to 3 layers of squamous epithelial cells. Yes, it is correct. It is differentiated or present between the inner enamel epithelium and stellate reticulum. Yes, it is correct. It is connected to each other and to the cells of stratum intermedium by desmosomes. Yes, that's also correct. It is essential for enamel formation as it contains acid phosphatase. This is the one which is wrong. Why it is wrong? Yes, definitely it is essential for enamel formation. Stratum intermedium function is enamel formation, not because it contains acid phosphatase, rather it contains alkaline phosphatase. So stratum intermedium contains alkaline phosphatase enzyme. So, and we know that alkaline phosphatase is essential for mineralization, enamel formation. Acid phosphatase is required whenever there is any bone resorption. Alkaline phosphatase is required whenever there is any bone formation. So stratum intermedium has got main role in enamel formation. So option D is also partly correct, but second half is wrong. Not because it contains acid phosphatase, but it contains alkaline phosphatase. A layer of cells that seems to be essential to enamel formation but does not actually secrete the enamel. Same thing they can ask you other way. Which layer of this is responsible for enamel formation but actually do not secrete? They just help is stratum intermedium. Actually, which layer secretes? Inner enamel epithelial cells. Inner enamel epithelial cells will become ameloblast and hence they will produce enamel. But the stratum intermedium is the one which is going to help this inner enamel epithelial cell to produce the or to secrete the enamel. The number of roots that are formed is determined by, so if you see, the number of roots is determined by the number of medial ingrowths that occurs at the epithelial diaphragm. We know that usually, right, the cervical loop will give rise to a extension. Suppose this is a cervical loop. The cervical loop will give rise to downward extraction called as HERS, and this HERS will form a medial growth. This will form a medial growth, which is called as diaphragm. Now, depending upon the number of diaphragm that forms, that decides the number of the roots. Okay, so root formation, I've told you that cervical loop will become HERS, and that HERS will further show medial growths, that is called as epithelial diaphragm. So number of this diaphragm decides the number of roots. Okay, let us go to the next question. Epithelial rests of Malassez are. So we know that what are epithelial cell rests of Malassez? These are the remnants of HERS. So these are remnants of HERS which are seen in the periodontal ligament. Okay, and they will undergo disintegration. These might undergo disintegration also. So if you see, all the three options are correct here. Epithelial strands and they may undergo degeneration. Epithelial remnants of HERS, network or isolated islands in the PDL, all of the above. So they are seen in the PDL. They are the remnants of HERS. They might undergo disintegration. Yes. See, basically, there is a question here. So can you please explain different coverings of newly erupted tooth? Yes, this is simple. Like basically you see there is something called as Nasmyth membrane. Nasmyth membrane is nothing but primary enamel cuticle. It's called as primary enamel cuticle. So what is this primary enamel cuticle? Primary enamel cuticle is like basically once a tooth erupts into the oral cavity, initially it will be covered with a basal lamina. That basal lamina is called as primary enamel cuticle or Nasmyth's membrane. So Nasmyth's membrane is just made up of the primary basal lamina. We have also called as the REE. We know that reduced enamel epithelium is there, right? But remember, the reduced enamel epithelium, once tooth erupts, it combines with the oral epithelium to form junctional epithelium, which is nothing but your epithelial attachment. So epithelial attachment, let it be primary or secondary. Primary, secondary depends upon what? Primary epithelial attachment, secondary epithelial attachment is nothing but your junctional epithelium. So that junctional epithelium that is formed once the tooth erupts into the oral cavity. So first covering will be, remember, Nasmyth's membrane after the tooth is erupted, and then the tooth will be covered by REE. That REE will combine with the oral epithelium and finally form the junctional epithelium, which is nothing but your attachment epithelium. Okay, I hope your doubt is cleared, Mehak. Tongue is not derived from. We know tongue is derived from different arches. First pharyngeal arch, third, fourth pharyngeal arches. Remember, second pharyngeal arch or branchial arch does not contribute for tongue formation. We know that two lingual swellings are there. One tuberculum impar is there, and we have one copula. So based on this, your tongue anterior 2/3 is formed and posterior 1/3 is formed. Okay, let us move to the next question. First evidence of calcification of maxillary permanent central incisor. This already we have seen that at 6th, 3rd to 4th month. Okay, in which of the following tooth distal marginal ridge is at a higher level than the mesial marginal ridge? What do you mean by higher level? Higher level means occlusal. That is called as the higher. Okay, so here in the mandibular first premolar, you will see this speciality, that the distal marginal ridge will be at a higher level or occlusal level compared to that of the mesial marginal ridge. So this is a simple memory based single liner MCQ that we should be aware of. Okay, let us move to the next question. The mesiolingual developmental groove is specific for mandibular first premolar. We know that maxillary first premolar has got mesial marginal developmental groove. This is speciality of or the uniqueness of the maxillary first premolar, whereas mandibular first premolar has got mesiolingual developmental groove. So we should be aware of these two grooves, which is the groove mesiolingual, which is the mesial marginal developmental groove. Okay, now let us move on to the next here about the next question. Membrana preformitiva. What do you understand by membrana preformitiva? Membrana preformitiva is nothing but a basement membrane basically, which separates the enamel organ and dental papilla before the dentine formation. It's not dental sac. So before that is till the early bell stage. Till the early bell stage, this layer, this membrane is known as membrana preformitiva, because this is going to separate the inner enamel epithelial cells from dental papilla. So membrana preformitiva is present till early bell stage. So in the late bell stage, this disappears, because once the hard tissue forms, this particular membrane disappears. This particular membrane disappears. Dentinogenesis imperfecta occurs due to disturbance in. Again, I have told you, we should be aware of the physiological stage of tooth development and the disturbance. That is a favorite topic. Whenever there is any different problem in the histodifferentiation, the dentinogenesis imperfecta can occur. Okay, because histodifferentiation is the phase where the inner enamel epithelial cell will become ameloblast, the dental papilla cell will become odontoblast. So because of this process only, you will see dentinogenesis imperfecta. Concrescence and enamel hypoplasia occurs in. So again, it's the same thing, memory based question. I've told you that it occurs because of apposition. Whenever there is any hard tissue formation, that is called as apposition. The last physiological stage is apposition, so during which the hard tissue formation occurs mainly. So concrescence, what is concrescence? Where the roots are fused, cementum is fused. Okay, two teeth, cementum is fused, that is called as the concrescence. Enamel hypoplasia, we know that enamel hypoplasia. So in both of these two, the basic problem occurs in the apposition. Basic issue occurs, basic defect that takes place is in the apposition. True statement among the following is: the length of the rods is greater than the thickness of the enamel. Yes, first statement is only correct. That means all the other three statements are wrong. The diameter of the enamel rod increases from the surface towards the DEJ. No, it increases from the DEJ towards the surface. Exactly opposite. Specific gravity of enamel is not 3.8, it is 2.8. The ratio between the inner and the outer dentinal surfaces is not 5:1, it's 2:1. Okay, so that is again difference here that you have to remember, that the first statement is only correct here. Then the length of the enamel rod is greater than the thickness. That is why enamel rods are basically, they don't have a straight, they have got a curvature. Okay, they have a curvature like this, enamel rods, they are not straight. Okay, dentine is formed in which stage of life cycle of ameloblast? The most important stage of life cycle of ameloblast is organizing stage. So in this stage only dentine formation occurs. Remember, this is considered to be the most critical phase of life cycle of ameloblast, because first dentine forms in this stage only. In this stage only. That is what is called as critical phase of life cycle of ameloblast. Okay, so we should remember the order also, very important. Life cycle of ameloblast order is very important. Always first comes morphogenic, next comes organizing, next comes formative, next comes maturative, next comes protective, last is desmolytic. Order is also very important. Weil's zone of pulp is again a simple single liner MCQ. The cell free zone of the pulp is known as Weil's zone, where you will not see any cells. Instead, it occurs in all stages. There is a question like epithelial mesenchymal interaction occurs in early bell stage or late bell stage. It begins in the cap stage, it increases in the early bell stage, it continues in the late bell stage. It occurs in all phases. Remember, in all the phases of the morphological stages of tooth development, you will see EMI, epithelial mesenchymal interaction. The cell-free zone is also, it contains mainly the neck plexus of nerves, that is called as plexus of Rashkow. Please remember, as it is not containing any kind of cells, it contains basically nerve plexus called as plexus of Rashkow. Which of the following are seen in non-keratinized epithelium? So understand here, the question is non-keratinized epithelium. So do you see keratinocytes? Do you see non-keratinocytes? Yes, both of them can be seen in case of a non-keratinized epithelium. There is some myth, it's a wrong thing that students usually understand, that non-keratinocytes are seen in non-keratinized epithelium. It's not like that. In any epithelium, you will see both keratinocytes and non-keratinocytes. Even in case of non-keratinized epithelium also, you will see keratinocytes and non-keratinocytes. Okay, gingival region, fatty zone, glandular zone are all seen in hard palate. We know that hard palate has got different zones: anterolateral zone, posterolateral zone. Anterolateral zone is nothing but fatty zone. Posterolateral zone is nothing but glandular zone, where it contains minor salivary glands. So we should remember that hard palate has got 4 zones: gingival zone, mid-palatine raphe, anterolateral zone, posterolateral zone. Anterolateral zone is also known as fatty zone. Posterolateral zone is also known as glandular zone. Which among the following is a keratinized area? If we see, gingival caul is non-keratinized area, junctional epithelium is non-keratinized area, lining mucosa is non-keratinized. So vermilion border of the lip is the one which is keratinized. Okay, we should remember vermilion border, also called as a red zone. It is also called as a red zone, remember. And this red zone is more, this is a zone where it is called as a red zone because more vascularity, and it also has got a protein, called as elidin. Because of both of these things, you call this as the red zone. Pristine gingiva refers to, what do you mean by pristine gingiva? The gingiva which is germ free. So actually, practically speaking, which is not possible. So in the experimental cases, they have seen that a gingiva which is totally germ free, bacteria free, should be called as pristine gingiva. Stratum germinativum refers to. So we know that the basal layer has got mitotic capacity, and the parabasal layer, that is just above that, has got the mitotic activity, which should be called as stratum germinativum. Because what is the meaning of germinativum? Mitotic ability, cell division ability. So basal cell layer, along with that the parabasal layer exactly above, has got the capacity to undergo mitotic division or cell division. Hence it is known as stratum germinativum. Odontoclasts consist chiefly of. Odontoclasts are the basically cells which are responsible for dentine resorption, and remember, anywhere resorption, it will be acid phosphatase. So there it contains acid phosphatase, that too the called as TRAP, tartarate resistant acid phosphatase. Joy, there is a question: if orthokeratinized and parakeratinized are types of keratinization, then why parakeratinized layer does not have stratum granulosum? I'm really sorry, you have understood wrongly. In parakeratinized layer, you will see stratum granulosum. It is not that you will not see. In orthokeratinized layer, it is prominent. Okay, it is in the Joy. Please understand that even in case of parakeratinized, you will see stratum granulosum. All the 4 layers are seen in stratum granulosum in the parakeratinized: basal, spinosum, granulosum, corneum. In orthokeratinized, the stratum granulosum is more prominent. Okay, I hope your doubts is clear. False statement about basket cell. Basket cell is the other name for myoepithelial cell. It is other name for myopia. That's true. It contains actin and myosin filaments. That's true. Its structure is similar to skeletal muscles. No, it is not skeletal muscle. It is similar to smooth muscles. It is similar to smooth muscles. They help in expelling the secretions from lumina of the secretory units. Yes, their main function is contraction. By contraction, they will expel the saliva into the ducts. So their structure is not similar to skeletal muscles. Their structure is similar to smooth muscles. Gubernacular canal and gubernacular cords are seen in relation to. They are always seen in relation to succedaneous teeth. What do you mean by succedaneous teeth? The teeth which are, they have a predecessor. Why not permanent teeth? Because succedaneous teeth are permanent also, but some permanent teeth do not have any predecessor. So they are not called as succedaneous. So succedaneous are the teeth which have got the predecessor. So there the gubernacular canal and gubernacular cord is there, and this is going to act as for eruptive guidance. So that will help in the proper eruption position of the eruption for the permanent teeth. That is the succedaneous teeth. Special stain for fat cell. We know that fat stains can be stained by mainly called the Sudan black. There is a stain called as a Sudan black. This is a stain that is used. Okay, there is one more stain, oil red O. Okay, oil red O, Sudan 3, Sudan black. All these are the special stains that are used for fat cells. So again, a special stains is again a very frequently asked MCQ. Chicken wire configuration is demonstrated by. In the basal lamina, particularly lamina densa, has got type 4 collagen. So this lamina densa has got basically type 4 collagen, which will have this chicken wire configuration. This will have this chicken wire configuration. Coming to the last question for today's session, dehydration of the specimen is carried out by. So we know that during tissue processing, we will use alcohol, that to ascending grade of alcohol. So that is the dehydrating agent. Okay, 10% formalin is a fixative. It is a fixative. Okay, nitric acid 5% is a decalcifying agent. It is a decalcifying agent. And liquid nitrogen we will use for basically the cryostat, frozen sections, for frozen sections. So we should remember that the increasing grade of alcohol, ascending grade of alcohol, like 50% first, then 60%, then 70%, then 80%, then 90%, then finally 100% alcohol, which is absolute alcohol. So in this direction, the tissue is moved. Initially it is kept in 50, 60, 70, 80, 90, 100. That is called as ascending grades of alcohol. So we do dehydration step to remove the water from the tissue during tissue processing. Okay, so I hope all of you have got some idea, some basic idea how to prepare, how to answer the MCQs. Okay, so best way always is to have a sound basic knowledge about the subject, then start solving the MCQs. Directly don't solve the MCQs. First go through the theory part of that. Let you follow the notes. Let you follow the textbook, whatever is that. Follow that and then start solving the MCQs. Okay. Thank you very much. Thank you very much, all of you, for the patient listening. Okay, and all the best for your exams. Any doubts, you can ask me any doubts later also, no issues. Okay. Thank you very much.